DNA connects to cellular respiration. The video is up for you to watch. Yeah. So I saw some cases yesterday. Today, our focus is just page 85 and 86. Our focus is page 85 and 86. While I prepare the whiteboard, could I get everyone to read page 85 and 86 if you have not? Okay, but I see some of you have already highlighted. You've already pre-read, knowing that today we're going to do this. Alright, now I think that's enough. Today we're covering DNA replication. Right, could everyone spend some time reading page 85 and 86? Not sure if we started recording already. No. Maybe I'll clear. Okay, we need your help here. Okay. Later on, later on. When you are done reading, find someone to talk to recap the structure of DNA. Then I'll ask some of you to share. Big about Spider-Man.
Just push it away. Just push it away. Let it bite us.
Yeah, man.
You got Spider-Man. Oh my god.
Bite me. Oh. It's not radioactive. Radioactive spiders always look very colorful.
It doesn't look radioactive. No, it doesn't. Yes.
Are you all done reading? Okay, sorry I distracted you all. Anyway, then really feel free to talk to your friends about it. of you are done reading. Okay, I'm going to recap where we left off last round. Remember last round I showed you a video of a small little salamander developing from a single cell. Okay, so let's continue from there. This whole chapter is called the cell cycle. You want to know what kind of a cycle of life that a cell goes through. Last week I showed you a pie chart of the general idea. Did I show you a pie chart? Right? No. Wait, were you absent? No. Oh, okay. Then everyone go to page 84. So I've not started at all. Okay. So, cells go through a cycle of themselves. They don't live forever. The skin cells that you have now aren't the same skin cells that were present maybe a few weeks ago. So we beckons the question, are you still you? Right? Are you still you? Right? We're actually replacing almost every single cell of your body every few months. The you when you were 4 years old and the you now, she's like totally different physical body. Right? We swapped out all the cells already by now. So are you still you? Oh, this is a philosophical question. Okay, so but cells go through a cycle. I'm going to just very quickly run through the steps of the cycle. Hey, have you started the video? Winnie, have you started? Okay, thank you. Okay, we begin first with that if you look into your books, you'll find that there is a phase called G1 phase. You see that? G stands for growth. So a cell has to grow, a cell does grow. Okay, G1 stands, all the G stands for growth. And then you find that there is S. S stands for synthesis. What exactly happens when the cell is growing? It grows a lot of things. More cytoplasm, more cell membrane, more organelles. What happens during the S phase? So it goes this way, the direction. What happens during the S phase is called S for synthesis. It synthesizes new DNA. Okay, the process is called DNA replication, which is our focus for today. DNA replication. After the cell divides its DNA, not divide, after the cell replicates its DNA, makes a copy of it, it goes through another growth phase called G2. Okay. Finally, a lot of cells, but not all cells, they go through this phase of cell division where they split from one into two cells. We will learn two forms of it in this following chapters. One is mitosis and one is meiosis. But that's not our focus for now. Okay? Together, G1, S, and G2, we call it the interphase. Because it's kind of like intermission before the highlight of the show, where the cell divides. Okay, so you realize the interphase is very, very long. The cell spends most of its life growing, making new DNA, before it divides into two. But actually, this is all still okay, but I have a question for you. Why in the world must a cell grow, produce double the amount of DNA, grow some more, before it can divide into two? Sorry? Okay, Javier says need to replicate, then I can divide into two. But then my question is, what happens if you don't do all this? You skip all this and you just divide. Like, is there an issue, right? Everything in biology happens for a reason. Why can't you further divide? Sorry? Okay, what if I say, when we divide, we often divide equally. So is there an issue? Why must the cell go through growth, produce more cytoplasm, more organelles, more cell membrane, more DNA, grow some more before it can divide into two? More space? More space? Or what, Alia? Aida?
Is it not the cell
Yeah?
won't even have enough things for itself?
Won't even have enough things for itself?
Yeah.
Yeah? Sorry, Nikita, what do you mean by space?
I mean, having cytoplasm and if it makes cytoplasm then it's
Oh, more cytoplasm, more room to put more stuff, is it? Okay. I get the feel that you are saying that we need to produce more so that we can share it at the end of the day. And that's the reason. If we were to let a cell just divide, when you get smaller and smaller and smaller and smaller cells, eventually the cell that has gone through many rounds of division, you end up with a cell with very little DNA, very little cytoplasm, very little organelle, and so on and so forth. So that's why cells need to go through this interphase that consists of growing as well as producing, replicating its DNA, so that I always have the same thing at the end of the day. Every time I divide, I'll have the same amount of what I started off with. That's the whole point of it. Our focus for the first part of this new topic, we're not going to focus on the growth phases. We're going to focus on the S phase. The S phase is extremely important. The S phase of interphase. Okay, we're going to focus our attention here. Our next two lessons, this one and the next one, is just about the S phase. Today we're going to talk about DNA replication. How do we replicate the DNA into two copies? The next lesson, we're going to talk about the experiment that was conducted that led to us finding out how DNA replicates. These two are the only things we need to know for the S phase. Right? And then after that, the final part of the chapter is learning about cell division, more specifically mitosis, and this thing called cytokinesis. And after that, we are done for your meetings. Okay? Yeah. Okay. So you see where we're going, huh? Is possible if you keep up and always constantly do your chapter stacks. We are nearly there. We only have one more chapter stack to go, right? Okay. So today our focus is DNA replication, and without even talking about the topic yet, which J3 topic you need to extract out from your brain? Biomolecules, more specifically, biomolecules, the chapter on nucleotides or nucleic acids, remember? Okay, so if there's a chapter you want to revise for this particular part, the S phase of interface, nucleic acids is extremely important. Okay, remember, I'm not going to ever assess you J3 topics separately as a question already. Always infused now. Okay, we assume that you have the foundation, and therefore, let's begin today with the structure of DNA. Okay, I'll give you some time to buzz with your friends. What are all the structures of DNA that you can think of? As many as possible. DNA structure, before I invite some of you to speak. I provided you a sample on the board for you to use. Okay, I feel like the bus has slowly fizzled out. Okay, Alia, will you give me one structure of the DNA you can think of? What about it? Oh, okay, what about nucleotides? Is Is it made up of nucleotides? Is it?
It's made up of nucleoside and phosphate.
Okay, made up of nucleoside and phosphate group. So actually, earlier just now you were telling me nucleoside is without a phosphate group, right? Nucleotide is the entire thing. Yeah. Okay. If we go back to our JH3 topic, recall the idea of a nucleotide. Okay. A nucleotide consists of a sugar group. It consists of a OH at the carbon number 3. It consists of a phosphate group at carbon number 5. It also consists of a base. Now they are saying that this is a nucleotide, right? Yeah. That's the building block of DNA. DNA is made up of nucleotide, but more specifically, it's made up of deoxyribonucleotide. So this is its building block. This is the monomer. This is what DNA is made up of. What is RNA made up of? What? Okay, just ribonucleotide, huh? Okay, so for RNA, RNA just consists, is made up of ribonucleotide. This is for RNA. This is for DNA. Okay, any more structural features? Shini? What's that? F sharp. Okay, yeah. Okay, there is 5 prime, 3 prime. The idea of directionality. Then when you look at the entire strand of polynucleotide, there is a 5 prime and 3 prime end. Okay, can you tell me where the 5 prime and 3 prime end is on this particular polynucleotide? 3 prime? 5 prime. What do you base it off? Phosphate group? Yeah. So notice phosphate group is here. The end with a free phosphate group is a 5 prime end. The end with the free hydroxyl group is a 3 prime end. If I flip it upside down, is it? Or flip it the other way around? Oh, kind of still the same, right? Okay, now we look at the bottom. This is the 5 prime end. This is a 3 prime end. And actually, how do we describe this feature of DNA? One run from 5 to 3, the other one run from 3 to 5. Okay, actually it's not complementary base pairing. The term we use is anti-parallel. The DNA is anti-parallel in nature. One strand 5 to 3, the other strand 3 to 5. I heard just now complementary base pairing. Complementary base pairing. How do we elaborate on that? What do you mean when you say complementary base pairing? Okay, only certain things can pair up. What can pair up what kind? Okay, A and T, C and G, complementary base pairing. But may I ask you what's the bond holding the AT, the TA, the CG? Good, you're still holding on to information. Hydrogen bonds. I have a question for you. If I were to join nucleotides together, for example, I take another nucleotide, I bond it to this nucleotide. I bond it to this nucleotide. What is the bond holding each nucleotide together? Yeah? What's the bond holding each nucleotide together so that I have this backbone? This bond does involve the phosphate group. So I'll give you a clue. The bond.
Phosphodiester.
Ah. Wow. That's a rule. Okay. The bond is the phosphodiester bond. So all these bonds in between the nucleotides, those are phosphodiester bonds. This is what holds the individual nucleotides together. But what holds two strands together are multiple hydrogen bonds, which is the stronger bond. Yeah, you're right. Phosphodiester bond is a stronger bond. Hydrogen bond is not really a bond where you share electrons. Okay. Any more structural features? Okay, one final thing. This is not so much a structural feature, but something I'd like you to hold on to. We synthesize DNA or RNA in the 5' to 3' direction. Okay, I'd like you to hold on to this piece of information in your head because this is an important feature of what we're going to talk about today. We synthesize DNA in the 5' to 3' direction. Okay, where's your board rubber? The duster. Okay, I found it. Okay, for those that don't quite know what it means when I say we synthesize DNA in the 5' to 3' direction, this is what I mean. I'm going to show it to you on a whiteboard now. Okay, so this is how DNA is synthesized, always in this direction. Watch out. This is the first nucleotide. Honestly, you have a choice of adding new nucleotides. Okay, assuming, this is the 3', this is the 5', right? Okay, what do I mean when I say DNA or RNA is always synthesized in the 5' to 3' direction? I mean this, You see, if I were to add new nucleotides to this end, I'm actually synthesizing it in the 3' to 5' direction. That's not what normally happens. What happens is this. DNA is synthesized in the 5' to 3' direction. So that's what I mean. 5 to 3. Okay, so hold on to that piece of information. I'm going to extend a bit more. This is what I mean by 5 to 3. This is the 5 end, this is the 3 end. We are synthesizing it in the 5 to 3 direction. 5 to 3 direction. Got it? Okay, so we're going to work with that piece of information now, moving forward. And hold it in your head. Okay, so now after reading the paragraphs, who found it like it doesn't make quite much sense? Okay, so when we look at our textbook, sometimes just words doesn't quite make much sense. So what we're here for today, we're going to try and make sense of what all the sentences, the paragraphs are saying. Right? And hopefully it has a little bit more meaning to you. First and foremost, I'd like you to identify all the enzymes involved in DNA replication. Okay, find all the enzymes involved. How do you know it's an enzyme or not? Okay, yes, we highlighted for you. Okay, so all the enzymes involved. We're going to find all the enzymes involved, try and make sense how we use them. Okay, ready? Number one, let's read the textbook, page 86. Okay, let's read the first paragraph. Proteins involved in the initiation of DNA replication recognize and bind to the origin of replication, causing the DNA double helix molecule to separate and open up to form single strands. This causes the formation of characteristic replication bubble. Okay, what does this look like? Okay, I'm going to zoom out now. Imagine this one. Each one of these strands I represent with a single line right now. Okay, what this statement is saying is this. The enzyme involved will come in and it will cause a region of the two strands to unwind and separate from each other. Okay, they are separated from each other to form what we call a replication bubble. Okay, this is a replication bubble. And the replication bubble is when the two strands in this region no longer hydrogen bonding to each other. So they're not very close to each other, look like a bubble. But this is where the heart of the action takes place. At the replication bubble, now that I've exposed the two strands of DNA, I can start using them as a template to create new DNA. Okay, I say again, now that I've exposed the strand over here, I can use it as a template to make new DNA. For example, I'm going to use this blue marker, I'm going to use this red marker to represent new DNA. What the enzymes are going to do is this. At the side of the replication bubble, it starts producing a new strand of DNA. Using the top strand and the bottom strand as a template. So I produce a new strand of DNA. That's how DNA replication occurs. I create many bubbles and I start building new DNA from within the bubble. How is the focus of today. We're going to talk about all the enzymes involved on how we're going to create new DNA. Are there only one bubble that will form? Actually, multiple bubbles will form. So imagine a case where I have a bubble just beside over here, forming the next part of the DNA. Here I, in a sense, create a new copy over here. So I have many bubbles, like mini classrooms that are building new DNA. Eventually, when the bubbles meet together, then we will just patch things up. Okay? So this is how DNA replications occur on a large scale. But we're going to zoom in today. On a large scale, we create many bubbles, and then we replicate the DNA in short segments. Then when the bubbles start to merge, we just join all the segments together to connect and form the new DNA. Okay, we're going to focus today on just one small segment. We zoom in right now. I'm going to zoom into this rectangle. I'm going to use this rectangle as where we're going to zoom into. Let's zoom into here. Okay, good. Anyone tell me what's the first enzyme involved in DNA replication? Helicase. Okay, so here's helicase. This is what helicase does. Okay, I'm going to draw helicase using blue over here. Helicase, it will be somewhere over here. What helicase does is to unwind the region of the DNA to form the replication bubble. So here's what's happening. Now we just focus on this portion here. So this is helicase. Helicase comes in and it starts to unwind the DNA. Starts to unwind the DNA. Starts to separate the two strands. Okay, so what I'm showing to you here is this region over here, just a small region. Yeah, I've just separated the DNA using helicase. A more important question is this. What bond is helicase breaking to separate the DNA? Hydrogen bond. Okay, so you must be aware of what bonds it's actively breaking to achieve this replication bubble. Okay, the next part I would like you to read and try to make sense of what's happening next. It's a little odd. Right, so the first two paragraphs, I kind of show you what just happened. The unwinding of the DNA double helix is catalyzed by helicase. Helicase binds and separates the double helix by disrupting hydrogen bonds between complementary base pairs, so that two single strands are available as templates. Okay, so now I have two templates. I have a template over here, template 1. Here's another template, template 2. So here's the tricky part of this segment. Let's try to make sense of this. Using the parental DNA strand as a template, parental DNA means these two DNA. Okay, using these fellas as a template, the enzyme primase adds about 5 to 10 ribonucleotides via complementary base pairing. This short strand of RNA is called primer, which serves as an anchor for deoxyribonucleotide to be added to the 3' end of the RNA primer. I'd like someone to come up and try to simulate what this is showing. Nikita, you try? Yeah, okay, come. Nikita, could you create this primer on this strand on top? Okay, so I'm going to provide you, Nikita, with ribonucleotides. Okay, because here are some ribonucleotides to play with. Which one will you use? Okay, here are some ribonucleotides. Nikita is primase, so she's primase now. Yeah, on my head. Okay, so she's primase. And the role of primase, they say, is to add about 5 to 10 ribonucleotides using complementary base pairing to form a short primer. How should we do this? Let's use this as the template strand.
Okay, so let's use 5 prime.
Okay, so it's 5 prime and 3 prime. It doesn't matter. What matters is we observe you how you create the RNA strand. Because we create new strands in the 5 to 3. Okay, so 5 to 3. Where will you add the next ribonucleotide? Okay, this is 5, this is 3, right? Okay. Okay, so Nikita now changed. She wants to build in the 5 to 3 direction. Is she doing it the correct way? Yeah. Okay. So hold on, Nikita. I'm just going to draw for you. This is the 5 prime end of the RNA that she's building. Notice she's adding it to the 3 prime end of the primer, right? She's building in that direction. So far so good. Is she using the rules of complementary base pairing? Yeah. Oh. Oh, there's no T. It's all RNA. Notice she has never used a T. Instead, oh, this is DNA. But there's an A here, right? So how? But then she therefore uses a U. Right? No T's. RNA doesn't have a T. You happy? Everyone happy, Nikita? Okay, shall we give her a round of applause? Yeah, okay, thank you. So, primase, that's Nikita. What Nikita is just this. She built this short strand of RNA in the 5 prime to 3 prime direction. We call this short strand, actually what is it called in the textbook? It's called a primer. Yeah? And it is made up of RNA. Okay, if it's not clear in the textbook, the primer is made up of RNA, which was what Nikita was trying to do for us. Okay, thank you, Nikita. Nikita has just built a primer for us. But what in the world is the primer for? Okay, let's remove helicase, give a little bit more space. Okay, for the subsequent part of this, I'm just going to make it, I'm going to remove some parts of the primer just so that we have more space. Okay, what's the next enzyme involved? DNA Polymerase. Okay, anyone wants to try to show on the whiteboard what is happening? What is DNA Polymerase doing? It says the addition of deoxyribonucleotides is catalyzed by DNA polymerase. Right? It adds to the 3 prime end of the primer. It adds new deoxyribonucleotides to the 3 prime end of the primer. So, Nikita has built a primer. What is the role of DNA polymerase? Anyone thinks that they have an idea of what's happening? Right, if you read the paragraph above, they say right after the bolder word primer, it serves as an anchor for deoxyribonucleotides to be added to the 3 prime end of the RNA primer. This primer here, we have just created an anchor where we can add deoxyribonucleotides to. That's what it's saying. That's the role of DNA polymerase. Arya, would you like to try? Okay, Arya is going to try to now build DNA. Okay, we just created an anchor, and now it's going to build DNA. Here are all your nucleotides you can work with. Now it's going to work with deoxyribonucleotides. Okay, you can use any of them, A, T, C, G. Okay. This is what Aryan doing making sense as we read the textbook. It says the primer serves as an anchor for me to add more deoxyribonucleotides to the 3' end. Yes?
Why do you need an anchor? Why can't you just add it straight?
Okay, so we'll answer that question now. Okay, thank you, Aryan. Okay, thank you. Let's give it a round of applause. So what has Aryan just done? He was DNA polymerase and he was adding new deoxyribonucleotide to this RNA primer. Then actually the first thing that will come to your mind is this. Why did I have to create this anchor to start adding new DNA? Right? Why not from the very start DNA polymerase comes in and start building new one already? That would be so much easier, right? Okay, I want to speculate why. Why can't we just start immediately?
You mentioned this last year.
I did. Oh, I did? I felt like someone asked me.
Yeah, someone did last year.
Yeah, somebody asked me. We just don't have the enzyme for it. Yeah? We do not have an enzyme that can start the synthesis of new DNA from scratch. We only have the enzyme present to add nucleotides to pre-existing 3' ends. Yeah? I say again, we do not have any enzyme that can add deoxyribonucleotides from scratch. We do, however, have an enzyme that can add deoxyribonucleotides to any free 3' ends that are existing. However, we do have enzymes that can add RNA nucleotides from scratch. So we play cheat. We use the RNA as an anchor for the DNA polymerase to serve this action. Okay? So I say again, we have enzymes that can add RNA from scratch. But we don't have enzymes that can add DNA from scratch. So it uses the RNA as an anchor to build DNA. Yes?
Then what about, let's say, just now there was no T, right? Because there was an A, but then there was no like T. Like during the primase section, there was A, but there's no T to complement it. So Nikita put in Q.
Yeah.
Then like, isn't that kind of wrong, right?
Okay, so Ida is saying, okay, you remember Nikita's primer went up to here, right? So let's say we build up to here. Okay, let's build the primer up to here, a longer primer. Okay, let's build a DNA from here, right? Let's build the DNA from here. T, G, and another G. Okay, so Ida, your question is, now this DNA is a bit weird. I actually have a U here, right? Right? It's a problem, right? Notice that with this system of synthesizing new DNA, the DNA will always have some RNA interspersed here and there. In fact, I will ever have a new DNA strand that consists of ATCG. I have some U's here and there also. I have a problem here, don't I? So what does our cell do? The final step of DNA synthesis is this. Let's read a little bit further. The very last paragraph below the diagram. They say in eukaryotic cells, there are different types of DNA polymerases. While one specializes in adding deoxyribonucleotides to the RNA primer, another specializes in removing the RNA primer and replacing it with deoxyribonucleotides. So this is how we solve the problem. Actually, we have two kinds of DNA polymerase. One that adds to the primer that was here. The other comes in later on to replace it. So I'm going to replace now. This DNA polymerase comes, it takes out the G, replaces it with a deoxyribonucleotide G. It replaces this G with another G. So it comes in later on to replace all of them. Do I solve the problem that Ida was talking about? Yeah? So this is the final enzyme, one of the final enzymes involved. I should replace with what? A. Okay? A, T and U shouldn't be together, right? I made a mistake. I made a mistake. It shouldn't be T and U. Correct? Like that? Yeah. Yeah? Okay, but you see I have a problem. Remember, DNA polymerase can only be elongated in a 5 to 3 direction. Over here, technically, what I've been doing here is okay. I was adding in a 5 to 3, 5 to 3, 5 to 3, 5 to 3. But this over here, right? To connect this to here, it's not the correct direction. I have a problem. Yeah? So actually, here to here, I cannot connect. Because DNA polymerase always builds in a 5 to 3 direction. If I were to add something here, it's building in a 3 to 5 direction. So how? We have one more enzyme. The last enzyme is called DNA ligase. DNA ligase helps to stick anything that cannot be stuck together. So it comes in, DNA ligase just comes in, and it forms a phosphodiester bond at the region where it couldn't form. It seals off all the gaps that couldn't be sealed off. That's the role of DNA ligase. Oh, will go away. This one is it? Is it? Okay. So Dania asked, after I synthesize the new strand of DNA, do I take it out after that?
It's like the top one and the bottom one is different, like the original.
So actually, what happens is this, I'm producing two double strand DNAs, right? So what happens is this. I ask you, which is easier? Which is easier? Is it easier for me to synthesize? Let me draw on the whiteboard. You tell me which is easier. Is it easier to do this? To produce two new strands and leave it like that? Or is it easier to, after you do this, you separate them and then you rejoin the new one and you rejoin the old one together to form two new strands? So which is easier? You think about it. Is it easier to just leave the new strand stuck to the template? Or is it easier to separate the new strand from the template and then stick the two new strands together and the two old strands together?
So we just break off of the original.
Yeah, so imagine I continue on building new DNA, then I'll have a new double strand. Bottom, I'll have another set building and I have a new double strand below. So that's how DNA replication occurs. I use the original templates to build new DNA, and then I now have two strands of two double-stranded DNA. Yes, Raksha?
So just now the part that you cannot, that's right. Why can you not do this?
Okay, so Raksha is asking, right, how come I cannot add this? So, okay, this is 5 to 3 prime, right? Am I adding the 5 to 3 prime direction? 5 to 3, right? Still okay? 5 to 3, elongating 5 to 3, elongating in a 5 to 3 direction, right? Now I have a question. Technically, this is the 5 prime end, right? Can you add nucleotides to the 5 prime end?
Oh, because that part is used to be the RNA.
Yeah, you cannot add new nucleotides to the 5 prime end. So this bond I cannot form the bond here, but ligase comes in to fix the problem. Yeah. Okay, so maybe you want to talk about this a little bit more. Do you understand this part? why I cannot join this to this over here. Okay, maybe I join out for you. This is a 5 prime, this is a 3 prime. It's a 5 prime, this is a 3 prime. I cannot edit in this direction. So this is where RNA ligase, DNA ligase comes in. Yes. I'm going to stop here for today. Yeah, I'm going to stop here for today to just take as many questions as possible. Yes. You truly understand what's happening, right? You realize that the bottom one is weird, right? Because I can build this direction, but I cannot build this direction, right? Yeah, right? Okay. I like it. Yes.
Why is it that we cannot add from 3 prime to 5 prime? Is it because the phosphodiester bonds are stronger than
from my understanding, we just don't have the enzymes for it. But I think in experimental, when we do it outside of our body, in experiment phase, right, we can actually build in other directions also. Yeah.
But then if you build it in other directions, then the phosphate backbone is no longer there, right? It's OH, right?
Oh no, it's not. So pictorially, don't go with the idea that the OH will remain. Yeah, it's just my paper. When I put it overlap, then it looks like OH is on top. Okay, let's spend some time reviewing. Okay, talk to your friends. Make sense of the entire process today. I will just take questions in the last 10 minutes or 5 minutes. Some of you will start to realize that there's a peculiar problem. If you understand what's happening on the whiteboard, right, you realize something is a little odd. For those that want to stretch yourself a bit, this is beyond the syllabus. Have you thought of whether this applies to the bottom strand? I'm going to show you right now. DNA, we synthesize it like that, right? Okay, let's build a primer at the bottom. Okay, so we see, I'm going to build a primer at the bottom. You see what you think about it. Let's put a C. Okay? So where should I put it? I'll put it like deeper in. Okay? Okay, your view is that way, right? Put it deeper in make more sense? Okay, let's put it a bit deeper in. Let's put it here. Oh, you're saying? I put it here lah. That's the 3 prime, this is the 5 prime.
Depends on where you're going to the next one.
Correct. No, it's okay wherever I put it, but the question is, where do I put the next RNA? Should I put it on this side or this side? Right? Okay, so 5 to 3, right? Okay, so I'm going to add the new RNA to the right side. So I need a what? A. Still okay? Okay, then I need a U. Okay, so let's say this is our primer now. Okay? Which direction do I synthesize and add the new DNA to? Do I add the new DNA on this side or do I add on this side? The right side? Okay, so let's add some more. So GGCC. Let's add G, G, C, thing? This strand on top, I can actually just go endlessly without interruption. Then this strand below, if I want to synthesize it in this direction, I cannot. Right? I cannot synthesize the bottom strand continuously in this direction like the one on top. How?
Then you put another. Then you just need to keep breaking it, and then
Keep breaking some more.
as long if it that dies, then it just stops.
What dies?
The helicase.
Helicase. Okay, so you notice, right, there's an issue, right? The strand on top I can synthesize in a continuous fashion. The bottom one, I put a primer here, then I synthesize like that. Then how? Oh, okay. She asked, is there helicase on the other end also? There is. Right, there is. Okay, but what if I tell you this? Actually, when the DNA is being synthesized, it actually synthesizes there is a directionality. They kind of synthesize on both sides, expanding out. Okay? So we look at the bottom strand. The bottom strand is a unique situation. We cannot synthesize the strand continuously this way. Right? We have to synthesize it this way, right? So we actually know from the bottom strand what the DNA, what the cell does is this. After it synthesizes this, it will just add another primer in front. You add another primer in front. So this is T. Let me add A. Let me add U. And let me add a C. And then it will add new DNA, which is what? I'm out. Okay, so pretend okay, pretend. Yeah, it will add some more. Then later on, it will just sub all this out and replace with new DNA. So there will always be one strand that will be built in segments, and the other strand can be built continuously. If you are curious about this process, so this is out of the syllabus. If you are curious about this process, go and read up on Okazaki fragments. Yes, it's Japanese. This person discovered this unique process. One side will be synthesized continuously, the other one is to be synthesized in parts. Okay, but I'm telling you this is out of our syllabus. You actually only learn it in the JC side. Okay, but if you're curious, you can go read out Okazaki fragments. If you are satisfied with today, actually the top one is enough. The idea of how we put primers in, we substitute it out, and then we seal it out at the end. Okay? Okay, thank you, Javier, for the question. Whitney, can we stop recording? I hope it recorded. For past few classes, he has been stopping halfway.